All right, watch and listen carefully here, guys. Number one, okay? Number one, this stuff here with the brown dots on it, the purple stuff is the rough ER, okay? So 1 mark for rough ER. And 1 mark for the fact that rough ER transports proteins and is involved in the production of hormones. Okay, either one of those functions is fine. They do not need to have both. Okay. All right, number 2, its counterpart, the smooth ER. Okay, so number 2 is smooth ER. Okay, and the smooth ER breaks down complex carbohydrates, builds and repairs membranes using lipids, detoxifies poisons. I'm missing something there. Complex carbohydrates, detoxifies poisons, makes lipids, repairs membranes. Sorry, I said the same thing twice. Okay, so any one of those functions would be acceptable there. You don't have to have them all. All right, number 3. What are we looking at here, this big purple thing? Nucleus. Nucleus holds your DNA. That's it, holds the DNA. Number 4, the purple dot in the middle of the nucleus, nucleolus. Okay, and the nucleolus copies DNA or controls protein synthesis. Either function will be fine. Okay, number 5, the outside of the nucleus is nuclear membrane. Okay, and I'll accept anything that has to do with keeping stuff out of the nucleus or containing the DNA in the nucleus or something like that. Okay, that's what its job is. Okay, what are the little dots everywhere? Ribosomes. What do? They make protein. Simple as that. All right, number 7, this big pinkish thing. Golgi apparatus. Okay, and the Golgi apparatus packages wastes for export from the cell. Number 8, cell membrane. And I'm looking for 2 words, endocytosis and exocytosis. It controls endocytosis and exocytosis. They must have those 2 words there or it isn't right. Number 9, the little sausage looking thing with the folds in it, mitochondria. Okay, and the mitochondria burn sugar for energy. And if they put cellular respiration, I would also accept that. All right, number 10, these little bubbles here that have stuff in them. Lysosomes. Okay, lysosomes break down food, digest food, site of digestion, something about breaking down food. Okay, number 12, the stringy things. In general, those are the cytoskeleton. Okay, but I will accept microfibers, microtubules, microfilaments, okay, as they're all kind of the same thing. Cytoskeleton basically supports the shape of the cell, supports the cell, something about that. Okay, guys, we keep our own paper till we're done. Okay, number 11. Okay, what are these 2 things here? Centrioles. Okay, job of the centrioles, they help with cell division. Give them a mark out of 24. Let them see it, and then I would like them in a nice neat pile right here, please. On your way up here, grab a Chromebook. Okay, so while you're getting set up there, okay, the purpose of these, and we do a couple of these throughout the year, of these literacy activities is to kind of show you how you need to read in science, okay? Because reading in English is different than reading in science, okay? Reading in English, you're like interpreting and trying to find hidden meaning and All that artistic stuff I don't understand. Okay. Whereas when you're reading in science, you're trying to find the nuts and bolts of what's going on in a particular article. A scientific article is written a lot differently than a piece of prose or a play or a story, okay, or things like that. So we have to read them differently, especially since usually when you're reading a scientific paper or something like that, you are trying to find information that goes along with something you're researching for, as opposed to reading, a play or a poem or something like that, where you're reading that for a different purpose. Okay. So what we need to look at is, how do we go about most efficiently getting the information that we need out of a scientific article? Okay. So one of the skills you want to have when you're reading a scientific article is that you want to be able to, let's say, identify key points. Okay. Now, lots of times key points are going to be in different font. They'll be italicized, or they'll be bold-faced, or something along, or underlined, or something like that. You always want to be looking for those. Okay. You also want to look at subject headings. Right? Usually a scientific paper is broken up into different headings. Okay. Okay, this section is going to be talking about OV deposition of rainbow trout in small freestone streams or something. Okay, like it'll be something, it'll have a topic. Okay, and if you were only looking at the swimming habits of rainbow trout, you probably wouldn't need to worry about how they deposit their eggs and their reds. Okay, in freestone streams, you could probably skip that part. Okay, it's probably not going to talk much about the mechanics of their swimming in that part. So you always want to be looking at, what am I looking for, and is this part going to have anything about that? If it doesn't, I probably will skip it. Okay, and that'll make you a much quicker and more effective reader. Here, put the phone away. As you're reading through an article, what else should you be doing? Sometimes it involves having something in your hand. Yep, highlighting is a good idea. Highlighting or underlining or whatever. Obviously, you're not getting a paper copy of this, but also making shorthand notes is a good idea. Okay, making your own kind of shorthand notes, which is kind of like highlighting. I started out in university as like the ultimate highlighter, right? By the time I was done with the textbook, it was like a kaleidoscope rainbow of different colors, because I had no idea how to highlight. I highlighted everything. It all seemed important. Okay, and so I would think, oh, I'm saving myself so much time, and then I'd start reading through everything I'd highlighted, and it was everything. Okay, so I would have like 4 different colors of highlighters. First, I'd go through with yellow and highlight everything. And then the second time through, I had a better idea, and I would highlight a little bit less, and the third time through a little bit less. Well, did that save me any time? Was that effective? Absolutely not, except that I reviewed several times. That was maybe good. Okay, but in the end, it wasn't efficient. It took me forever to read stuff and get what I needed from it. Okay, what I should have done was look ahead to what I was supposed to get first. Okay, if you're reading an article and you're supposed to answer some questions on it, read the questions first. I mean, I know that seems like common sense, right? But a lot of us don't do it. Okay, we just, oh, well, I'll read the article and then I'll read the questions, and you're like, oh, I forget everything I read. Okay, if I know what the questions are asking, I know what to look for. Okay, I'm going to highlight or make shorthand notes on the right stuff. Okay, or I'm going to say, oh, right, paragraph 4. That's where the answer to that question is. Okay, things like that. Always know what you're looking for before you start reading. Otherwise, you're just reading for reading. Okay, so we've got this article that's on reverse osmosis. Okay, which is an artificial process, but works similar to how real osmosis works, except in reverse. Okay, real osmosis is always trying to find a balance. Water's moving across a membrane and making salts equal in concentration on both sides. Reverse osmosis is forcing water through a membrane and leaving all the salts on one side. Okay, and then having clean water on the other. So rather than achieving a balance, it's achieving a complete imbalance, but it's also using energy to do it, where regular osmosis is passive. Okay? So we're going to be looking at that. Okay, looking at that article, but what we want to do first is, what are we going to be asked about? Okay, so these are the 4 questions you're going to be asked about. They're in the Google Doc file that I attached there. Okay, first question, compare and contrast the membranes used in reverse osmosis with a cell membrane. How are they similar? How are they different? All right, first thing I'm writing down here, look for stuff about membranes. Okay, that would be my first note to myself on a sheet of paper. Okay, beside my computer, I've got look for stuff about membranes. What are membranes made out of? Okay, that's something I got to look for. Right, so that's one of the things I'm going to make a note of when I come across it. Okay, I got to mark that paragraph and make sure I get back to it. Okay, Second question, what provides the force needed to move water across the membrane in both osmosis and reverse osmosis? Okay, explain why one process requires energy and the other is passive. Well, we've already talked about how regular osmosis works. Okay, so we kind of already know that, but we should look for something that explains how osmosis works. So I would write another note that says, movement across membrane, how accomplished, or something like that. A little jot note to myself, I got to look at that, okay, in both. Okay, maybe write something about force, and then how would I make something go across the membrane? Okay, if I want to push something through a membrane, I'm probably going to need a what? What kind of force? What kind of a device would create a force with a fluid? Pressure making, how do I make pressure? I need a pump, right? Sorry, I was just expecting that one. Okay, anyway, so we probably need a pump of some kind. I might make a note, look for stuff about pumps. Okay, something like that. These are things that I want to look for to help me answer question number 2. Okay, question number 3. In a short paragraph, explain and justify if you think using reverse osmosis would be an acceptable way to conserve water. All right, what does conserve mean? Right, use less, right? So I've got to make a note again that says, all right, does reverse osmosis use less water? Okay, does it save water? That's something I got to look for. Does it or doesn't it? I better find out in the article. It does say in there somewhere, okay, whether it uses more or less, or how much it wastes if it wastes any, or how much does it conserve if it conserves it. Okay, so I would have a note in there that says something about, does it save water? Okay, so would it be an acceptable way to conserve water? It doesn't say anything else, just conserve. Okay, so we're not looking for whether it can purify water. Obviously, it can do that. Okay, that's what reverse osmosis was developed for, was to purify water. Okay, in fact, the process was initially developed by NASA to make sure that they could recycle their urine. Okay, on a spacecraft. Okay, over long periods of time. Okay, it could filter all the salts out of the urine and create usable water again. Okay, things like that. All right, and then it was mentioned in the article for question number 4 that reverse osmosis was used to ensure impurities were removed from certain products like wine and syrup. Okay, so it's being used to take stuff out. All right, can you think of any reasons why a purist might disagree with the use of this process? What's a purist? Right, someone who wants it done the old-fashioned way, or wants their food natural, or things like that. Because originally, winemaking was you put yeast and grape juice and sugar in a barrel, and you leave it there for a while. Okay, and then eventually you make sure it's sealed airtight. And the yeast will use up all of the oxygen in the barrel, and there won't be much of it there, and they'll start carrying out a process called fermentation, okay, which makes, of course, alcohol, right? And that's what turns grape juice into wine. Okay, is this process that the yeast carry out. So you put yeast, the stuff you use to make bread rise, in grape juice, and then you drink it after. So they use reverse osmosis to make sure that all the gunk that forms on the bottom of a wine barrel gets taken out. Otherwise, you get chunky wine. Okay? You laugh. Really expensive wines have sediment on the bottom of the bottles. Okay? Yeah, they will. Okay, that's the remnants of the fermentation process. A lot of them will still have that stuff on the bottom. Okay, there is sediment in some of them. Okay, so why would a purist disagree with removing that stuff? Okay, well, maybe that's something I should find out. Maybe it doesn't really say anything about the article. Maybe I've got to read the article and kind of figure that part out for myself. Okay, those are things that I want to look for as I read through the article. Okay, everybody follow me there? All right. What's a major stumbling block for understanding if I'm reading a scientific article? Wording. Yeah, exactly. What if I don't understand all the words? Well, I have to look them up, but I mean, it can certainly make it difficult, right? We consider somebody to be a proficient reader if they read with 95% effectiveness. That means they understand 95 out of 100 words. Now, you read a 1,000-page book. Well, yeah, you've missed out on, like, by the time you're done, you've missed out on 50 pages. You didn't understand 50 pages of the book by the time you're done. Well, that's not good, okay? But we consider that to be proficient at reading, 95%, okay? Mr. Kennedy always talks about this with band. He actually played for us last year what 95% sounds like in band. It was god awful. It sounded terrible. He's like, see, you guys are all happy with 95 in your science classes, and look what it sounds like in band. And we're like, oh, makes me feel awful. Okay, but yeah, I mean, you think about it, we got to read at a much higher rate than that, especially in scientific articles, in order to be effective. So, while you're reading through this article, write down words you don't understand. Okay, if you don't know their meaning, just write them down, and then in a few minutes, I'll go over any words that come up. Okay, so don't be afraid to ask. Write them down. If you don't understand them, chances are lots of other people didn't either. Okay, unless of course it's like the. Okay, don't ask about the. Okay, but if there's a word you don't understand, write it down and we'll go over them. So for right now, I'm going to give you a few minutes and I want you to read the article. Okay, and remember, you're looking for certain things to answer those questions. You might want to be writing some things down physically on paper or going back and forth to the document I gave you and typing things in there as you go. Okay, that's the process you're going through right now. And then in a few minutes, we'll go over the words we don't understand. If we got any terms that we are in doubt about. Desalinization? Okay. This one? Okay. Yeah, so desalinization is any process that removes anything dissolved in the water from it. Okay. One easy way to desalinate is to distill water, that is to boil it off, and then condense the vapor back. That leaves all the salts and stuff behind, but reverse osmosis will desalinate water as well. Okay. Other ones? Don't be afraid to ask, guys. Okay. How many people are done reading the whole article? Okay, well, I'll give you a little bit more time, and if more come up, we can go over them. Okay, so natural osmotic pressure is simply the pressure that is created when there's an imbalance of salts. So if I've got a whole bunch of salt on one side of a membrane and not very much on the other, that's going to create an osmotic pressure that pushes water from where there's no salt to where there is salt. That's what causes osmosis to happen, is that imbalance creates pressure. That's what moves the water. Right, yeah. When you have that diagram that's in there, there's osmotic pressure. Right, because the salt can't get across the membrane, only the water. Right, so the water moves to achieve a balance. Right, the water to salt ratio on both sides will be the same, even though there's going to be less fluid on one side than the other. Yeah. Okay, any other ones? Okay, so we're looking for pretty thorough answers, guys, on that, because I am going to be taking that in for marks. Okay, on the 7th of April, you still have about 10 or 11 minutes left in class, so you can get quite a bit of that done. We may have a little bit of time on Monday after Easter to work on this, but certainly not the whole class. Okay, so you don't have to do it over Easter. Okay, your cell anatomy lab, I think I may do the Wednesday, right? Okay, so. Okay, Tuesday. So make sure that obviously you come in and see me on Monday after Easter, okay, with questions or whatever on that one. Okay.